Good day, teacher. My name is José Roberto Moreno Vargas, a student of 10th grade A, and I'm going to talk about you the importance of the microscope in medicine, health, and general science. Importance of the microscope today. The importance of the microscope in medicine, health, and science in general is due to the fact that it is a tool that allows observing cells, particles, bacteria, and microbes, among other organisms and elements that would be invisible to naked eye. The arrival of the microscope produced a revolution in the way of thinking of the human being, through which the body and its affections began to be studied in a scientific way, starting from meticulous observation of it. Today, taking advantage of advances in technology, microscope enable the detailed study of cells and molecules, among others, allowing specific research on drugs and disease. Reasons for the importance of the microscope. Studies of organisms, particles, and microorganisms. Since it was invented, the microscope has helped to study organisms and particles invisible to the naked eye, whose existence was not known. This has allowed the creation of new areas of study, both in biology, medicine, and science. On the other hand, there are different types of microscopes useful in various fields of study, such as medicine, health, and natural science. Be able to perform precision medical surgeries. Surgical microscopes are used to perform surgeries in different medical specialties, during which, due to delicate tissues to be intervened, the surgeon needs to increase his vision. In this way, the manipulation and repair of a large number of systems, such as veins, blood vessels, and nerves, is more precise and better results are obtained. Study of viruses and molecular structures. The cryo-electron microscope allows to obtain atomic precision when making observations of macromolecular structures and nanometric structures, without the need to use a large amount of sample volume. Another field where it is used for frequently is that of medicine, allowing the three-dimensional construction of the parts that make up different types of cells. It is also a useful tool for studying viruses such as HIV, facilitating the development of effective treatments for their eradication, based on its understanding and careful analysis. Disease investigation. This type of microscope is characterized by creating an electron beam, which is directed so that it impacts on a tissue sample to be observed, and we passing through it generates a detailed image of it. The image magnification scale is about 100,000 times that of the original sample size, allowing in this way the visualization of the interiors of cells and identifying DNA molecules, chromosomes, and atoms. Materials used for our experiment. We only need a little water, a large syringe without a needle, adhesive tape, some support, a laser pointer, a clothespin, and a wall to project on. These elements must be mounted in such a way that the light from the laser point falls on a sample suspended in a drop of water, as in the example shown in the image. Thanks for your attention.
Hi teacher, good morning. My name is Carlos Arístides Rivas Calderón, and today I'm going to present you, I'm going to be explaining how it works and how it is made, the homemade microscope. First, we need to know which materials we will need. First, we need a syringe. Then we need a needle, and we need two objects that are in the same height. For example, 2 glasses of water and a laser. Well, the process is we need to put both glasses of water and we need to put the syringe between those glasses so that the syringe can be upside down. After that, we need to take our needle and and carefully touch our tongue or walls of our cheeks. Why? Because we need the germs of our mouth in the needle. After that, we need to heat the, no. After that, we need to take the syringe and push a little to make a single bubble on the top. So after that, you take your needle with your hairs of your mouth and heat or yeah, you need to heat with the needle the bubble of the water. And after that, you need to be you need to be in a place that is dark because in light you can see the light of the laser, but in the night you can see it. For this, you need to take the laser and put it in a position so that when the late light of the laser hits the bubble, this can make a projection of germs of our mouth. For example, you can see in that image. And then why this projection you can see clearly? Well, I have here a little explanation. When a light, for this example of the laser, hits the little bubble, this light hits the wall of the bubble so many times that this light finds the exit and show us a projection of the germs so much bigger. In other words, when you hit the bubble with a laser, with the light of the laser, this laser inside the bubble hits all the walls of the bubble so many times in so many directions that it makes a force that this finds an exit, and with the refraction inside of the bubble it makes everything inside much bigger. In this example, we can see the gems of our mouth so much bigger, like you see in the past image. Well, thank you for your attention. I hope you understand me. I hope you understood this project that is easier and interesting. Have a nice day.
Hello teacher, my name is Victor Sebastian Garcia Lucero. And so today I'm going to talk about types of bacteria that can be found in the mouth. Well, first of all, the mouth is made up of many surfaces, each of which is covered by a large number of bacteria. Some of the bacteria in the mouth play a role in the development of diseases, such as tooth decay and periodontitis. Both are risk factors for developing other more serious diseases, such as diabetes, mellitus, and cardiovascular diseases. The composition and concentration of the bacteria in the mouth depends on several factors, such as nutrient availability, temperature, oxygen concentration, anatomical features, exposure to immunological factors. What are bacteria in our mouth? Bacteria in the mouth, along with other microorganisms, are known as the oral microbiota. That population is not fixed, but is continually changing, even due to factors as simple as yawning, kissing, or eating certain foods. In general, aerobic and anaerobic bacteria predominate inside the mouth, both gram-positive and gram-negative. Among these, Lactobacillus, Actinobacillus, Staphylococcus, or Streptococcus. Facultative anaerobic gram-positive cocci predominate in saliva, representing around 44% of the bacterial population. It is followed by strict anaerobic gram-negative cocci, which account for about 15%. The gram-positive facultative anaerobic bacilli have a similar percentage. Factors such as tooth loss, and diseases such as gingivitis, alveolaritis, or periodontitis can produce changes in the composition of the saliva microbiota. Tobacco use and porogene also play a role. Where are they found? Buccal mucosa. The following types of bacteria predominate in the buccal mucosa: firmicutes, mainly of the Streptococcus and Paeonella genera, Protibacteria, especially Neisseria, bacteroids, Parabotella, and Actinobacteria, Micrococcinae. Glagine of the oral mucosa prevents colonization by Treponema denticola and Fusobacterium nuclearum. According to some studies, bacteria on the oral mucosa could be involved in some types of cancer. Bacteria in the teeth. If the teeth do not have cavities, the following bacteria are usually found there: Campylobacter, Granulicatella, Kingella, Leptotrichia, and Streptococcus, especially Streptococcus sanguinis. Likewise, Haemophilus parainfluenza, Gemella aeromolaisans, Slackia exigua, and Rothia species are present, especially in adults. The teeth are surfaces that facilitate the formation of biofilms. Such biofilms are changed based on different factors. Some bacteria such as Streptococcus mutans, Actinomyces, and Lactobacillus affect tooth decay and periodontitis. Gums and bacteria in the mouth. A biofilm can also form on the gums, which can cause diseases such as gingivitis. If the gums are healthy, microorganisms such as Proctobacteria predominate, in the particular the Groma Protectobacteria of the genus Actinobacter, Haemophilus and Moraxella. When there are health problems, Streptococcus granulicatella and Gemella are also found. Treponema denticola is present in the superficial part of the gum biofilm, along with Porphyromonas, Gingivitis, and Tannerella forsythia. Likewise, there may be viruses and sometimes microorganisms such as fungi. Oral diseases modify bacterial communities. Bacteria can also be found on the tongue. The biofilm that forms on the tongue is also dynamic and harbors many of the bacteria in the mouth. Approximately 45% are facultative anaerobic gram-positive cocci, especially Streptococcus salivarius, followed by Streptococcus mitis, miliary group of Streptococcus cocci, and Streptococcus mucilaginus. strict anaerobic gram-negative Cauchy and facultative anaerobic gram-positive bacilli are also found. To a lesser extent, various species of the Lactobacillus neisseria, Fusobacterium, and Haemophilus genera can be detected. Fusobacterium nuclearum, Porphyroma gingivalis, and Taenera forsythii have been found on the back of the tongue of people with halitosis. How to prevent pathogenic bacteria from the mouth. Oral hygiene is the key to preventing pathogenic microorganisms that affect our oral cavity. In the same way, frequent visits to the dentist may make it possible to identify risk factors and incipient pathologies that can be stopped before definitive damage.
Good morning, good afternoon, or good night, teacher and classmate. Today I'm going to talk about the purposes of our project. Okay, this project arose from our desire to encourage more people to study and investigate their surroundings using a microscope as an instrument. We choose this project to solve the lack of this instrument in people who want to deepen or estrange their knowledge in the field of chemistry and biology. In both cases, the use of this instrument is very important for the study of different microorganisms, such as cells, bacteria, viruses, etc. We consider that the implementation of this project is of the utmost importance, in addition to being totally affordable for everyone. And another objective was observe small objects is better detail. Will a microscope with accessible materials develop interest in the structures of objects that are not very visible to the human eyes, and finally provide ease in the study of biology and chemistry. Thanks for your attention. Have a good day.
Welcome, teachers. The name of our project is Home Microscope to see germs in the mouth by Diego Marcelo Gonzalez Aguilar. Introduction. To begin with, how big is the cell? There is no single answer to this question because there are more cells than cells, and some are larger than others. Almost all of them are microscopic, but some are so large that they are clearly, even inevitably, visible without the microscope. Initially, man saw the urgency of solving the most basic needs. He was creating the tools that allowed him to transform reality and begin a civilizating process that has not stopped in its evolution. From the discovery of fire, the invention of tools, and the development of language, to the most recent developments that have allowed the disintegration of the atom and the use of atomic energy, the exploration of the cosmos, and the development of multiple disciplines or fields of knowledge, such as the biotechnology, nanotechnology, and infotechnology. The human being, a thinking subject, endowed with the intelligence, memory, and will, has made numerous contributions. Over the years, science has progressed and evolved, changing the lifestyles that people to the point of turning a dream into a day-to-day action. It is impressive how we can adapt the most composed object into a simple instrument. This is the case that we will talk about how why and structure of one of the most important instruments in science, the microscope.